Welcome back, everyone. So we're going to continue on with Chapter 6, Reproduction at the Cellular Level in our OpenStax textbook. So in this section of our textbook, we're going to get into cancer and the cell cycle. So we've already learned about the cell cycle, so we're going to talk about how we can have errors that occur, which can lead to uncontrolled cellular division, which leads to cancer. So by the end of this chapter, you should be able to explain how cancer is caused by uncontrolled cell division, as well as understand how proto-oncogenes are normal cell genes that, when mutated, become oncogenes. You should be able to describe how tumor suppressors function to stop the cell cycle until certain events are completed, as well as explain how mutant tumor suppressors cause cancer. Okay, so again, we're basically going to learn about cancer, and it's really not that complicated. You might think cancer is this complicated disease. Well, it's just the result of unchecked cell division, and that's just caused by breakdown of the mechanisms that regulate the cell cycle. Okay, so that usually begins with a change in the DNA sequence of a gene that codes for one of those regulatory molecules. And so when we have, faulty instructions, it can lead to a protein that doesn't function as it should. And so if we have a disruption to the monitoring system, it can allow other mistakes to pass to our daughter cells, and then those daughter cells now have those mistakes that will pass to the daughter cells of the daughter cells. And so each successive cell division will give rise to daughter cells with even more damage. And so that leads to these cells that are end up being cancerous. So we'll talk about that in this video. So what is cancer? So cancer is caused by uncontrolled cell division. And again, it's due to mutations. So what are mutations? So mutations occur during DNA replication. Okay, so when does DNA replication occur? So that occurs during the S stage of interphase. So remember, during interphase, the cell replicates its entire genome. And that's a lot of nucleotides that it has to replicate. And so there is a chance that there will be an error, and that during the G2 checkpoint, that the error will go unnoticed. Okay, so if it goes unnoticed, it can end up staying, and when the mitosis takes place, that error will be passed to the daughter cells. And in that particular gene, what do genes do? They code for proteins. So that gene can code for a malformed protein. And so we end up with this malformed protein. So that malformed protein could lead to uncontrolled growth of a mutated cell. And so those mutated cells then continue to grow at a pace that is faster than normal cells. Okay, so they end up growing more often and at an uncontrolled rate than our normal cells. And so what ends up happening is you get a clump of mutated cells where you shouldn't have any cells. And so that forms a tumor. Okay, so you've probably heard of tumors. So tumors, they can be benign, they can be malignant. So a benign tumor is just a clump of cells. They're malformed cells. But they tend to be a little bit more slow growing, or they tend to not spread. So those usually are not as worrisome as a malignant tumor. So a malignant tumor, they tend to be more spreading. So they tend to break off and spread. And if these cells spread into your bloodstream, then they can travel to other areas of the body, and then settle into other areas of the body, where then they continue to divide and grow more tumors, and then that leads to cancer or uncontrolled cell division. Okay, so we're going to start off by watching an Amoeba Sisters video on the cell cycle, which will kind of talk about, will be a review of the cell cycle, since we talked about that in the last video, as well as what happens when we have this uncontrolled cell division that leads to cancer.
Have you ever been sitting in class and thought to yourself, I wonder what my skin cells are doing right now at this very moment? This kind of pondering may be unique to me, maybe, but wouldn't we at some point wonder what our cells are doing right now? Because if you remember, as part of the cell theory, you are made of cells. All living things are made of one or more cells. Many multicellular organisms like you have cells that work together, working together as part of a body tissue. Body tissues working together as part of an organ. Organs working together as part of an organ system. Your cells are specialized to work in these different levels of organization. You have skin cells, stomach cells, muscle cells, just to name a few, and their functions need to be regulated. These cells actually are regulated as part of something called the cell cycle, and that is going to relate to my question of, I wonder what my cells are doing right now. Cells themselves can grow in size. But let's put it in perspective now. A multicellular organism isn't growing because each individual cell is getting bigger. A multicellular organism itself grows by making more cells, by the cells making more cells by dividing. That's cell reproduction. One reason that you're bigger than you were when you were 5, unless you are 5, is because your cells have divided to make more cells. Mitosis and the cytokinesis that follows, that splits the cytoplasm, allows you to make new body cells. But you don't want that cell division happening all the time. Why? It is likely that you've heard the term cancer before. We have had family members that have battled cancer before. It is definitely a relevant topic for all of us. Cancer is in part due to cells that divide too frequently. The cells are not regulated. They are uncontrolled. Cancer cells can have other problems too. They might not be able to communicate with other healthy cells. They may not be able to carry out normal cell functions. They may not securely anchor themselves like other cells do, which can make them more likely to travel somewhere else. Some cancer cells have the ability to secrete their own growth hormone that makes blood vessels divert over to those cancer cells and supply the cancer cell with nutrients, which can take nutrients away from healthy cells. Why do cancer cells become this way? Well, there is a lot of research in this area. With some cancers, there may be genetic links, making some cells more susceptible to having problems. These genetic factors might run in families. Exposure to toxins, radiation, excessive exposure to UV light, all of these can be risk factors for some cells to become cancerous. The uncontrolled growth that cancer cells have gives rise to more cells like them, which can develop into a tumor. Some tumors stay put, but some do not. Now, the good news is that scientists continue to develop better treatments, which include destroying the cancer cells with radiation or medication, such as chemotherapy, which will target cells that divide frequently. Maybe someday you will be part of helping to meet the challenge of trying to eliminate cancer, because the fact remains that these cells are not participating in the cell cycle like they should. So what is the cell cycle? The cell cycle is often represented as a pie chart like this. Cells are either in one of two different phases. A phase called interphase, where the cells themselves are growing, replicating their DNA, doing their cell functions, or they are in M phase, which includes mitosis and the actual splitting of the cytoplasm, cytokinesis. So this M phase is where cells actually divide to make more cells, but cells spend most of their time in interphase. So most of the time, they're not dividing. Now, depending on what kind of cell, it might do mitosis more or less often. For example, your hair follicle cells do mitosis frequently, which is why your hair can grow at the rate that it does. It's also why many cancer drugs may also target hair follicle cells, because many cancer drugs go after cells that do cell division frequently. It's a big deal for cells to hit this M phase. If a cell has an error, a harmful mutation for example, you do not want it to divide because then it will create another cell that has this same issue. That's where checkpoints come in handy. Along the cell cycle, there are checkpoints to check that the cell is growing well and replicating its DNA correctly and doing everything it's supposed to do correctly before it divides. To better understand those checkpoints, let's further divide this cell cycle pie chart. We have G1, S, G2. All three of those are part of interphase. Then we have M phase where mitosis will happen. During G1, the cell individually itself grows. Then it replicates its DNA in S phase. You can remember that because the S is for synthesis, which means to make something, and it's making DNA. Then G2, the cell grows some more in preparation for mitosis. So let's take a look at checkpoints. We've got one here in G1. This checkpoint checks, is the cell growing well enough? Is its DNA damaged? Because if it is, you definitely don't want it to move on to S phase where it would replicate DNA. Does the cell have the resources it needs if it were to keep moving on? This checkpoint in G2 checks if the DNA was replicated correctly back in S phase. Is it growing well enough? Does it have the resources it needs to continue? Okay then, moving on. This next checkpoint in M phase is my favorite checkpoint. It checks in the stage metaphase to make sure the chromosomes, which are made of DNA, are lined up in the middle correctly, that they're all attached to the spindle correctly, because if they're not, the chromosomes will not be separated correctly. So now you may have two big questions. First, what happens if the cell doesn't meet the requirements of the checkpoint? And second, what is doing the regulating of this cycle anyway? To address the first question, if the reason the cell can't go past the checkpoint is a reason that can be fixed, the cell may kind of pause here until it can fix that issue. But if it can't be fixed, then the cell does something called apoptosis, which basically means the cell self-destructs. This ensures that a cell that is damaged beyond repair will not go on to divide. So what is doing the regulating anyway? We've mentioned before that proteins are a big deal. Genes in your body can code for proteins that do an assortment of functions, and there are many proteins involved with regulating the cell cycle. Some of them are positive regulators because they allow moving forward in the cycle, and some are negative regulators that might make things stop. The proteins themselves can be sensitive to cues inside and outside of the cell. So, for example, two proteins that are involved in positive regulation are cyclin and CDK. CDK is specifically an enzyme protein, a fancy kind called a kinase, which is worth a Google. CDK can have different forms of cyclin protein bound to it. Different types of cyclin rise and fall throughout the cell cycle, and the rising and falling is based on a variety of signals to determine when the cell should move on to the next cell cycle phase. Typically, each cell cycle phase, G1, S, G2, M, will tend to have a different cyclin binding with the CDK. The rise and fall of cyclin types and the role CDK has when it's active is a fascinating subject to explore. Remember that vocabulary word we said, apoptosis? Proteins that are negative regulators, for example, a protein called P53, can be involved in initiating apoptosis. Again, we encourage you to explore beyond the video. One last thing to mention. There are some cells that don't go through the phases we mentioned because they're actually in G0. That's a 0, by the way, and not a no, because if it was a no, then it's a go, and G0 is kind of the opposite of that. G0 is a resting phase. Now, cells here are still performing cell functions, but they're not preparing to divide. Some cells go here temporarily, maybe if there's not enough resources around, for example. But some, like many types of neurons in your brain and spinal cord, may stay here permanently. If they stay here permanently, they'll never get to M phase, so they will not divide. This can be one reason why a major injury to the brain or spinal cord can have challenges with healing, as many of those cells may not be able to replicate, a topic that definitely continues to be researched. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so that video was a great kind of a refresher of the different parts of the cell cycle and the different checkpoints. as well as an introduction into those positive regulators and those negative regulators. So we're going to get into some of those, that information that was introduced, starting with basically what regulates the cell cycle. So, how do we know when a cell is going to go through cellular division? Well, we do have, genes that control cellular division, and they regulate the cell cycle. So those genes are called proto-oncogenes. So these proto-oncogenes are normal genes that actually control cellular division. Okay. Now, we can have genes that these proto-oncogenes, they can actually become mutated. And, there can be an error that occurs during cellular division, which isn't repaired, and then that error becomes, part of the daughter cell. And if it happens on a proto-oncogene, it will then become an oncogene. So an oncogene is basically a mutation of a proto-oncogene where you have an error. Okay, so this error, now being an oncogene, allows for uncontrolled progression of the cell cycle. Okay, so instead of the cell, getting the signal of when to divide, it basically always gets the signal to divide. Okay, so it never gets the no signal. It continues to divide over and over and over again. Okay, so proto-oncogenes are supposed to regulate the cells when they're supposed to divide, but when they become mutated, that goes away. Okay, so then they're constantly in the on position. Okay, so think of it like a light switch. Okay, or like a light switch. So proto-oncogenes are in control of the light switch. So when the light switch turns on, that means the cell can divide. When the light switch turns off, that means the cell cannot divide. But if there's a mutation in this gene, that makes the light switch constantly be turned on. And so once the cell divides, it will then divide again and again and again and again and again. Okay? And so that leads to cancer, because you have the cell dividing over and over and over again. And so you end up with this clump of cells, a tumor, and then that clump of cells, ends up, leading to cancer, which is that uncontrolled cellular reproduction, excuse me. Okay? So proto-oncogenes, think you have the light switch that turns on and off. Oncogenes, it's constantly on. Okay? So that, again, is kind of like a positive regulator, turns the cell either will go into the cell cycle or not. A tumor suppressor gene, that's considered a negative regulator. So it's a gene that codes for regulator proteins that prevent the cell from undergoing uncontrolled division. So the video alluded to one of them, so P53. A couple examples of others would be the protein retinoblastoma, or RB1. And then there's another one, P21, that's more common example. So like P53, if you have P53 turned on, so that's going to basically cause the cell cycle to arrest, and that will signal this. If the cell is damaged, then that will tell the cell to undergo programmed cell death, which we know as apoptosis. Okay, so apoptosis is just a fancy word for cell death or cell suicide. So the cell will self-destruct. Okay, so it knows that something is wrong with it. Its DNA inside is damaged, and so it needs to undergo programmed cell death. Okay. And that's due to that, the DNA is damaged, the cell cycle, there's cell cycle abnormalities, there's hypoxia, for example. So that P53 tumor suppressor gene will turn on, and it will basically tell the cell to undergo programmed cell death. Okay. If everything looks okay, the cell can repair, perhaps instead of undergoing cell death, it could repair itself, and then the cell cycle will restart. Okay. So basically this protein, it can stop the cell cycle, and if the damage is repairable, it will repair itself. If the damage is not repairable, the cell will undergo apoptosis. Okay, so this is what tumor suppressor genes do. Okay, so they will help the cell repair damage to their DNA or tell the cell to commit suicide. Okay, so that's for the normal process. Now, if we have a mutation in our tumor suppressor genes, which can happen, okay, so we can have some sort of damage to our tumor suppressor genes, and so we can code for an abnormal P53 protein. So if we have an abnormal P53 protein, then none of this happens, and so the cell cycle continues. And if the cell cycle continues, then the DNA repair never happens, and so we end up having the cell continue to divide with that mutated DNA strand, and so we have the proteins malformed, and then we have the tumor cells continue to divide and divide and divide, and then we get the uncontrolled division leading to cancer. Okay, so the mutated P53 protein does not arrest the cell cycle, so we end up with the damaged cell continuing to divide. Okay, so this is an example of a negative regulator, so it will prevent the cell from continuing to divide either through cell death or through having it repair itself. But if we have a mutated tumor suppressor gene, it will allow the cell to continue to divide, becoming cancerous. All right. So those are two ways that we can have uncontrolled cell growth. So we have an error in our tumor suppressor gene leading to uncontrolled cellular division, or we can have, if we go back, an error in our proto-oncogene, which regulates whether a cell goes into the cell cycle or not. Okay. So we're going to finish off with a video from Khan Academy. So Khan Academy is another one of those places that you can go if you're not understanding one of our biological concepts. You can always look up that concept with Khan Academy, and they do a great job. So this one's on cancer, and again, your book doesn't really go into much detail about cancer, so that's why I wanted to show you this video, which kind of goes into more detail about what cancer is, and so that, because everybody has, either a friend, a family member knows somebody who has struggled with cancer, so this gives you a little bit more understanding of what it is.
Most cells in the human body just go about their business on a daily basis in a fairly respectable way. Let's say that I have some cell here. This could be maybe a skin cell, or really any cell in any tissue of the body. And as that tissue is growing or it's replacing dead cells, the cells will experience mitosis and replicate themselves, make perfect copies of each other. And then those two maybe will experience mitosis. And then if they realize that, gee, it's getting a little crowded. There are other cells in my neighborhood. They'll recognize that and say, I'm going to stop growing a little bit. That's called contact inhibition. And so they'll just start going, and then let's say one of them experiences a little defect, and he says, you know what, gee, I'm not really, something's a little bit wrong with me. I, the cell, recognize this in myself, and the cells will actually kill themselves. That's how the good of cellular citizens they are. They'll kind of make way for other healthy cells. So this guy might even kill himself if he realizes that there's something wrong with it. There's actually a cellular mechanism that does that. It's called apoptosis. And I want to make this very clear. This isn't somehow some type of outside influence on the cell. The cell itself recognizes that it's somehow damaged and it just destroys itself. So apoptosis. So that's the regular circumstance, even when there is a mutation. And just to give you an idea, even if mutations are relatively infrequent, and I don't know the exact frequencies at which mutations occur, I suspect it's of different frequencies in different types of tissues, there are on the order of 100 billion, let me do a different color. They're on the order of 100 billion new cells in the human body per day. Per day. So even if a mutation only occurs 1 in a million times, you're still dealing with roughly 100,000 mutations. And maybe most of the mutations, maybe they're just some little random things that don't really do a lot. But if the mutations are a little bit more severe, the cell will recognize it and destroy itself. And I want to make a very clear point here. I'm talking about the cells of the body, or most of the body. This could be the cells in my eye, or the cells in my brain, or the cells on my leg. These aren't my germ cells. So these mutations, even if the cell survives, will not be passed on to my offspring. That's an entirely different discussion when we talk about meiosis. These are all my body cells, and they're replicating, and we've gone over this with mitosis. Mitosis. So any mutations here, they'll either do nothing, or the cells might malfunction a little bit, or the cells might hurt themselves or hurt me, but they're not going to affect my offspring. I want to make that point very clear. Now you're saying, hey Sal, 100 billion new cells a day, that must mean like every cell in my body is recreated. Well, and that just gives you an idea of how many cells we have. We actually have on the order of, it's obviously not an exact number, but there's actually, in the human body, there's on the order of 100 trillion cells. 100 trillion cells. And if you look at it that way, you say on average 1,000 of your cells replicate each day, but the reality is some cells don't replicate that frequently at all, and some cells replicate much more frequently. But this just gives you, just to take a little side note here, this gives you an appreciation, I think, for the complexity of the human body. And we think of our own our world economy and world society is so complex. It's made up of 6 billion humans. We're made up of 100 trillion cells. 100 trillion. Let me rewrite 100 trillion in billions. 100 trillion can be rewritten as 100,000 billion cells. And each one of those 100,000 billion cells are these huge, I shouldn't use the word huge, but they're these complex ecosystems in and of themselves with their nucleuses, and we'll talk about all the different organelles they have, and we talked about cellular replication, DNA replication, and how the cell replicates. So these things aren't jokes, and they have all of these, these complex membranes that take things into them, and they are creatures to themselves, but they live in this complex, you can almost view it an environment or society that is each of us. So that's just a side note, just to appreciate how large and how complex we are. But you can imagine, and this is how I got off on this tangent, if we're making on the order 100 billion new cells every day, you're going to have a lot of mutations, and maybe some of the mutations, I said some of them don't do anything. Some of them, the cell recognizes that the cell is just going to be kind of dead weight, so the cell kind of eliminates itself. But every now and then you have mutations where the cell doesn't eliminate itself, and it also deforms the cell. So when you have that, let's say I have some cell here. I have some cell, and it's got some mutation. I'll do that mutation with a little X right here. With a little X. That's it's in a DNA. Maybe it's got a couple of mutations. So one of the mutations keeps it from experiencing apoptosis, or destroying itself, and maybe one of the mutations makes it replicate a little bit faster than its neighbors. So this cell, through mitosis, it makes a bunch of copies of itself. There are a ton of copies of itself. And this kind of body of cells that essentially has a defect, they're all from one original cell that kept duplicating, and then those duplicating. But all of these are defective cells. If you were to look at them in kind of a, compared to the tissue around it, it would look abnormal in some way. Maybe it wouldn't function properly. This is called a neoplasm. 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 Now, a lot of neoplasms, well, they don't have to form a body like this. Sometimes they might somehow circulate in the body, but most of the times they form this this big lump, and if they get large enough, they're noticeable, and that's when we call it a tumor. So if this is actually a lump of kind of differentiated of tissue that's definitely abnormal, that's what you call a tumor. So the term neoplasm and tumor are often used interchangeably. Tumor is the word we use more in our everyday vocabulary. Now, if this lump just kind of grows to a certain size, but it doesn't, it's just there, doesn't really do anything dangerous. It's not replicating out of control. I guess it's not replicating a lot faster than its neighboring cells, and it's just hanging out, maybe growing a little bit, but not in any significant way harming our environment. We call that a benign tumor or benign neoplasm. Benign. And benign essentially means harmless. Benign tumor. That means if it's, that's good. You want to hear that. If you got a lump, God forbid you have a lump either way, but if you do, if it's a benign tumor, that means, that lump, it could kind of stick around, no damage done. But if these DNA mutations, maybe some of these it is benign, but maybe one of the benign ones has another mutation in it that starts making it grow like crazy. Grow like crazy. And not only does it grow like crazy, but it becomes invasive. And invasive means that it doesn't care what's going on around it. It just wants to infiltrate everything. So let's say that guy grows like crazy, and he starts, let me do it in a different color, and he starts infiltrating other tissue. So he's invasive. Invasive. So super growth. He's invasive. So he doesn't care what's going on. He's all of a sudden turned into some type of a cellular psychopath. And even worse, his descendants, it's not just one cell anymore. He just keeps duplicating and passing on this kind of a broken genetic information that makes it want to replicate. And then maybe even more, there could be more and more things that break down in its offspring or the DNA that comes from its replications. And actually that's a good likelihood, because the same parts of its DNA that broke down, some of the DNA that broke down in this guy, some of the mutations might have actually hurt the DNA replication scheme, so that mutations become more frequent. So more frequent mutations. More frequent. So as these replicate, more and more mutations appear, and then maybe eventually one of the mutations appears that allows these cells to break off and then travel to other parts of the body. And then in those parts of the body start to take over and start taking over all of the cells. And this process is called, the cell has this is one of the hardest words for me to say, something wrong with my brain, but the cell has metastasized. Metastasized. You might have heard the word metastasis, and that's just the notion of these run amok cells all of a sudden being able to travel to different parts of the body. And I think you guys know what we call these cells that aren't respecting their cellular neighborhood. They're growing like crazy. They don't experience that contact inhibition. They're invasive. They start crowding out other cells and hogging up the resources, and they keep mutating really fast because they have all of these genetic abnormalities, and eventually they might even break away and start infiltrating other parts of the body. These are cancers, or cancer cells. Cancer cells. And so you might have an appreciation for why this is so hard. Cancer is such a hard disease to quote-unquote cure, because it really isn't just one disease. It's not like one type of bacteria or one type of virus that you can pinpoint and say, let's attack this. Cancer is a whole class of mutations where the cells start exhibiting this fast, invasive growth and this metastasis. So, you might look at one type of cancer and be able to say, hey, let's target, the mutation where the cells look like this, and you're able to knock out some of them. Let me do a this and this color, so maybe you're able to knock out that guy, that guy, that guy. But because their DNA replication system might be broken in some way, they continue to mutate. So eventually you have one version that's able to not be knocked out by whatever method you get, and so you have this kind of new form of cancer, and then that new form of cancer is even harder to kill. So you can imagine that cancer is it's kind of a never-ending fight, and you kind of have to attack the general idea behind it. When you chemotherapy and radiation, all of these type of things, they try to attack things that are fast growing, because that's the kind of one common theme behind all of the cancers. And we could do a whole playlist on what cancer is and how people are attacking it. But I wanted to at least show you in this video that cancer really is just a byproduct of broken mitosis, or even more specifically, broken DNA replication. That we have all of these cells replicating themselves every day, on the order of 100 billion, and every now and then something breaks. Usually when they break, the cell, either nothing happens or the cell kills itself, but every now and then the cells start replicating, even though they're broken, and sometimes they start replicating like crazy. If they just replicate, but they're really not doing any harm, it's benign. But if they start replicating like crazy, taking over resources and spreading through the body, you're dealing with a cancer. So hopefully you found that interesting. You already know a good bit of the science that kind of deals with what is probably one of the worst ailments that we deal with as creatures. And obviously we're not the only people who can experience cancers. Even plants have cancers.
Okay, so again, I like to show that video just because it gives us an understanding of what cancer is, which again plagues all of our families and friends and loved ones. So again, from that video, we see what cancer is, and if we bring it all together, we can see that cancer, being uncontrolled cell growth, is caused by mutations from the proto-oncogenes that become oncogenes, or tumor suppressor genes, mutations in that. Those are a couple of causes that can lead to cancer. Okay. So let's do a couple of review questions. Blank are changes in the nucleotides in a segment of DNA that codes for a protein. So is it proto-oncogenes are changes in the nucleotides? Tumor suppressor genes are changes in the nucleotides, gene mutations are changes in the nucleotides, or negative regulators are changes in the nucleotides in a segment of DNA that codes for a protein. So what is it called when we have changes in nucleotides in a segment of DNA that codes for a protein? If you said gene mutations, I would agree with you. Okay, so a simple gene mutation is a change in the nucleotides in a segment of DNA. So remember, our DNA is just, that segment of nucleotides, the A's and the T's and the C's and the G's. So if we have a change in that, that will, genes, they code for proteins. So if we have a change in this, that will form a malformed protein. So a mutation is just a change in that sequence. Okay. Another question here. A gene that codes for a positive cell cycle regulator is called A or N kinase inhibitor. B, a tumor suppressor gene, C, a proto-oncogene, or D, an oncogene. So what is it called when we have a gene that codes for a positive cell cycle regulator? So what gene is that? So if it's a positive cell cycle regulator, that would be a proto-oncogenes. Remember, proto-oncogenes, they're the ones that turn on the, they're the light switch, right? So they will turn on, give the go signal for the cell to enter the cell cycle, or, say, no, you're not ready. So it's the positive. The tumor suppressor gene, that's the negative regulator, right? Okay, so the gene that codes for a positive cell cycle regulator is the proto-oncogene. If that mutates and causes it to constantly be on, that would be the oncogene. All right, so that's it for our third section in Chapter 6, Cancer and the Cell Cycle. So by now you should be able to explain how cancer is caused by uncontrolled cell division. You should also be able to understand how proto-oncogenes are normal cell genes that, when mutated, become oncogenes. You should also be able to describe how tumor suppressors function to stop the cell cycle until certain events are complete. Okay, so remember that, like, for example, we use the example of the P53 protein. So if that one will either allow the cell to repair its DNA. If the DNA is cannot be repaired, then the cell will undergo apoptosis. And then finally, you should be able to explain how mutant tumor suppressors cause cancer. Okay, so if we have a mutation in the tumor suppressor genes, a mutant P53, then we end up getting, no repair take place, and then the cell undergoes uncontrolled cell growth. All right. So that is it for 6.3, Cancer the Cell Cycle. So in the next video, we're going to switch gears. We're going to focus in on prokaryotic cell division. So that's a little bit different. We're not going to talk about mitosis anymore. So it's very simple, prokaryotic cell division. It's basically through binary fission. So we'll talk about that in the next video.